Please do hurry and join the live class so that we can start the live session. Before we dive into today's session, make sure to subscribe our YouTube channel Edwins if you have not subscribed already, so that you can never miss the live classes and new educational videos. And also an important announcement for the upcoming board students: you can purchase our premium version with a very low price, where you can get the unlimited study materials, mock test series, and unlimited educational recorded videos. So please do visit and purchase the premium version. And please do hurry to join the live class so that we can start the live session. A very good evening to all of you and welcome to the another live class of Edwins. In previous live class we have discussed about the different motivation or locomotion in animals, and today's class we will discuss from the second chapter, the cell division and cell cycle part 1, where we will discuss about the cell structure and its division methods. So what is cell division? Cell division mainly, here I have draw a figure, that is the mother cell and two daughter cell. So cell division is the process from one cell divided into two daughter cells, 2 or 4 maybe. So these are the types of cell division, but primarily from the mother cell the daughter cell are produced. This is called the cell division. So for the cell division, cell need to grow and develop and divide the cell materials or cellular materials. So what are the cellular materials? We know that cell contain two parts. One is, if this is a cell, this will be the nucleus. This is a cell. This is the nucleus. Okay, so the cell is surrounded by, all plant and animal cells are surrounded by cell membrane. Okay, so this cell membrane, inside the cell membrane, this part is known as protoplasm. Protoplasm. So this protoplasm contain this matrix part as well as the nucleus. So protoplasm is consist of cytoplasm plus nucleus. Okay, so nucleus is the cell organelle which is important part of a cell, which contain the hereditary substance, that is gene. Okay, we will discuss it, what is gene, what is the function of gene, or where it is located, we will further discuss. But for now, that cell have two main parts, that is cytoplasm and nucleus inside the cell membrane. So these cytoplasm contain several types of cellular organelles like mitochondria, Golgi complex, ribosome, and protein, lipids, etc., etc. So they all have to be divided and contain twice to divide in two cells. So the cellular contain need to be 2x so that it can give rise the two similar cells. So for this context, the cellular part also divide, and as well as nuclear part or the nucleus inside the nuclear material should be divided into double. So this nucleus contain the special and hereditary character, that is the chromosome. So we will discuss about the chromosome first. So what is chromosome? Chromosome is the Chromosome is the ribbon-like or thread-like structure. Which contain gene. Okay. So this chromosome is a thread-like structure which contains gene, that is the characters of an individual being. So the gene which carry the characteristics, different kinds of characteristics like eye color, hair color, skin color, height, etc. Different types of characteristics it bears for an organism or for an individual. So this is the chromosome. So what is the structure of chromosome and what is the morphology of a chromosome? These chromosomes are mainly two types according to their function. In human body, the chromosome are two types according to their function. So what are they? Number one is autosome, and another one is sex chromosome. Also this is known as allosome. Okay, we know in case of human there have 23 pairs of chromosome. How many? 23 pairs mean 46 chromosomes. Number of chromosome is 46. Number of chromosome is 46. But in case of allosome or sex chromosome, there have 22 pairs in autosome and only one pair of sex chromosome or allosome is present. So the autosome in case of human is 22 pairs or 44, and one pair of, that is 2 allosome or allosomes are present in human body. What are they? In case of female, this is XX, and in case of male this is XY. So these are the two types of chromosome present in our body, which is, this is mainly the sex chromosome or allosome found in the reproductive cells, and the autosomes are found throughout the body. These chromosomes are also found in somatic cells or body cells. Okay, so these are the types of chromosomes in our body. So what is the primary structure of a chromosome? The primary structure of a chromosome I have drawn here, that is here I have drawn a chromosome. This chromosome have two parts. If this is a giant or macro figure, in micro figure the chromosome is like this. This part is this part and this part is this part. Okay, and chromosome, before going to the structure, first that chromosome have two arms. One is P1, another one is Q1. The short arm is known as P1 and the long arm is known as Q1. Okay, so P is the short and Q is the long. So these chromosomes, these two hands, here is the central region, and these two hands is known as chromatid. Okay, this is the chromatid. Now the question is, this is the structure of chromatid. This part is highly condensed of genetic material which is known as DNA. Okay, what is DNA? We will discuss further, but for now the structure of chromosome, this part is known as satellite, one end. So the satellite is the ending part or the telomere part of a chromosome. Telomere part is the ending part of a chromosome. Okay, so the ending part of a chromosome which is known as telomere, and this telomere when structure like a bulb or knob, this is known as satellite, and this satellite containing chromosome is known as sat chromosome. Sat chromosome. Okay, so this is the satellite containing chromosome is known as sat chromosome. This sat chromosome, next one is the part that is chromonema. So before going to the structure of chromonema, the DNA actually present in two form, or the genetic chromosome is present in two form. That is, one is like this spiral and another one is simply coiled. This part, okay, this red part, simply coiled. This spiral part is known as, the spiral configuration is known as plectonemic coil, and this simple or the loosely compact is known as perinomic coil. These are the chromonema. So before the chromonema, the DNA, the chromosomes are arranged like this. This is the, we know that we receive the chromosome, one from our mother and one from our father. So if this is the father and this is the mother, so this structure, if I draw here, this structure is so like okay, like this. Okay, if magnify this one structure, if I magnify this one structure, it will look like So this is the structure of a magnifying structure of this one part. This is known as chromomere. Okay, so this highly condensed and backbone structure of chromosome is known as chromomere, and this is the chromatid. These both are chromatid, and this is known as chromatin knob. So this is the structure of this part, this highly condensed part. This is the part of magnifying part of this. From this to this, and here from this to this part. If this is 1, this will be the 2, this is the 3, and this is the 4. The highly or super coiling part of the chromosome. Okay, so this is the chromomere, and this highly condensed part of the chromomere is known as, or bead-like part is known as chromonema, or in case of plural, this is known as chromonemata. Okay, this is in singular, but in plural this is chromonemata. In case of plural. Okay, this is the chromonemata. So the structure is clear. This is chromomere, is the this part of the chromosome is known as, the bead-like part of the chromosome is known as chromonema. Now the region that is primary constriction, where the two chromatids are get attached. This part is known as, also known as centromere. What is centromere? This part where the two chromatids are attached. This part is known as centromere. Okay, where two chromatids are get attached. This centromere region have contain a special protein which is known as kinetochore. This part of the chromosome in centromere region here have special protein which is known as kinetochore. What is the function of kinetochore? Kinetochore help in the division of sister chromatids in cell division. Okay, for now the function, that is kinetochore is a special protein which help in the cell division to get divide to sister chromatids. These are mainly a hook-like structure which attached with the spindle fiber and get separate to sister chromatids. Okay. Now the part is, that is pellicle. That whole part of the chromonema is covered with a thin layer which is the covering of it, which is known as pellicle, and inside the pellicle there have a jelly-like substance which is known as matrix. And the another region, that is there have another constriction beside the primary constriction. These are known as secondary constriction, and these secondary constriction are also known as secondary constriction, also known as NOR or nuclear organizer region. Okay, this is also known as nuclear organizer region. So what is the function or why it is located? Nuclear organizer region is mainly contain the repetitive sequence of ribosomal DNA. This is known as tandem repeat. When a sequence is overlap or repeated cycle, this is known as tandem repeat. So these tandem repeats is responsible for protein synthesis. Okay, protein synthesis. This protein will help in several functions of the body, like the chromosome 15, 14, 21, 22. These chromosomes, this number of chromosomes are contain these non or tandem repeats of ribosomal DNA. So this is the basic morphology or morphological structure of a chromosome. Now the chromosome contain the genetic material. So what is the genetic material or what material it will carry? The genetic material is the DNA and RNA both. So these, the chemical composition of chromosome. Okay, so the chemical composition of chromosome, here chromosome have two parts. One is protein and another one is nucleic acid. Okay, these proteins are also present in two forms, that is histone and non-histone. And this nucleic acid have two forms, one is DNA and another is RNA. Okay, so in protein these histone are present in 5 forms, that is H1, H2A, H2B, H3 and H4. These are present in 5 forms. And non-histone proteins like tyrosine, tryptophan are present. In case of nucleic acid, they have two forms, that is one is DNA, which is stand for deoxyribonucleic acid, and RNA stand for ribonucleic acid. Okay, so this is the whole part or chemical composition of the chromosome. So how it is organized? the backbone part of the chromosome is the DNA, and DNA is like a ladder-like structure which was proposed by the Watson and Crick, and this is known as Watson and Crick model, where they have proposed that DNA is a ladder-like double helical structure. It has two strands. So this is the DNA. This DNA will wrap upon the histone and it will form the histone protein are present number of 8. And these 8 histone protein wrap upon the DNA. This DNA wrap upon the histone protein, and this structure of this wrapping upon the histone protein is known as nucleosome. So what is nucleosome? Nucleosome is the structure of histone octamer, that is number of 8 histone protein are wrapped by the DNA, which is known as histone octamer. This histone octamer or nucleosome further condensed into like this structure, which is known as chromatin fiber, and this chromatin fiber further condensed or coiled and form like this, which is known as chromatin. And from this chromatin, the supercoiled part of the chromatin is chromosome. Okay, the relationship between DNA and chromosome is clear. DNA wrapped upon the histone octamer, which is known as nucleosome, and these nucleosomes and rest part of the DNA condensed to form the chromatin fiber. This chromatin fiber further condensed or supercoiled into form the chromatin, and these chromatin are finally coiled and form the chromosome. So chromosome is the supercoiled version of DNA. This is the relationship between DNA and chromosome. So this DNA structure, before going to this DNA structure, the important part, that is the chromosome have two parts, the highly stained and normally stained or lightly stained. The highly stained structure and this normally stained structure is basically occurs due to the condensation or the coiling of the DNA. So the the chromosome is divided into two parts, that is heterochromatin and euchromatin. Heterochromatin is the part where the supercoiling is occurred or supercoiled part, and this part does not take place or participate in cell division. Do not participate in cell division, and this is highly stained. In the time of cell division, if I fixed a cell in this the cell division condition and stained, this part will be stained highly, and in euchromatin this is light stained part. And it is participate in in cell division. Okay, and in case of human, 92% genome is present in the euchromatin region. So this is mainly the active region, this is mainly the inactive region of the chromosome. So I am talking about the genome. So what is the gene or what is the genome? Gene is the specific part of our DNA. So gene or genome is the specific, if this is the DNA, this is the DNA. So all part of the DNA are not active like the chromosome. So few parts like this part is active, but this part is not active. So this active part can synthesize protein. Of course, the DNA cannot synthesize the protein. From the DNA, it produce RNA. This RNA can produce the protein. So this part can encode, more appropriate term is encode a specific protein. Which part is encode protein? This part is known as gene. And our DNA have several parts that encode the different kinds of protein, which is entirely called the genome. So genome is basically in the form of plural, gene is the singular. So we are talking that human genome project, the whole gene or the whole genome or the genes are present in human is categorized or enlisted in the human genome project. So this is the DNA, and according to the model of Watson and Crick, the DNA is have a specific structure as well as it is contain three main parts. In DNA, it have three parts. Number one is pentose sugar. What is pentose sugar? That is 5 carbon containing sugar molecule, of course organic sugar molecule. Next one is the nitrogenous base, and the third one is the phosphate. Okay, so these are the three parts of the DNA, and these three are collectively called nucleotide. And this nitrogenous base and pentose sugar, this nitrogenous base and this pentose sugar are togetherly called nucleoside. How you can remember this? Tri, from the tri means 3, from the tri and tide. T stands for 3 and side stands for 2. Okay, so this is nucleotide and this is nucleoside. Pentose sugar and nitrogenous base is nucleoside, and pentose sugar, nitrogenous base and phosphate togetherly called nucleotide. This, in case of RNA, these three are common, but this pentose sugar is replaced by ribose sugar. Okay. In case of RNA, this pentose sugar is replaced by ribose sugar. So here he has a term that is nitrogenous base. So what is the nitrogenous base? Nitrogenous base is categorized into two categories according to their structure. What are they? The nitrogenous base is categorized into two types. One is purine and another one is pyrimidine. The purine are the double ring structure and the pyrimidine is single ring structure. Okay, so purine are the two, that is adenine and guanine, and pyrimidine are also two types, that is thymine and cytosine. Okay, so these are the two types of four types of nitrogenous base present, that is categorized into two category. One is purine and another is pyrimidine. In purine they have adenine and guanine, and in pyrimidine that is thymine and cytosine. This is in case of DNA. But in case of RNA, all are present, this adenine and guanine, but this thymine is replaced by uracil. This thymine is replaced by uracil, and the pairing between this purine and pyrimidine is like this. Adenine and guanine are joined with double bond, and this guanine and cytosine is joined with adenine and thymine, and this guanine and cytosine are joined with triple bond. And in case of, this is for RNA, in case of DNA, in case of RNA, this is A, T is replaced by uracil, and guanine and cytosine as it is. So this is the structure. So now the how it's look like DNA. If this is a pentose sugar, here is a phosphate molecule, and here is the nitrogenous base. Here is also another pentose sugar, the phosphate molecule, and a nitrogenous base. So this is the 5 carbon where the phosphate molecule get attached. Okay. Now the number 3 carbon is joined with the next one phosphate. Okay. There is O. This is the 5. So this, like this, the chain is formed. But this is the left side, but in the right side this is opposite or mirror image. How? In the right side it is look like this, inverted. And here this part is T, this is C, and this is T, and these are the parts where phosphate get attached. Okay, so this is the ladder form or the form or model is proposed by the Watson and Crick. Okay. And in case of RNA, this is only single strand. In case of DNA, this is double strand. Number 1, this strand and this strand. But in case of RNA, this one strand is removed. Only one strand is present. That's why they are not ladder in form. They are present like this. They are joined together like here is A and here is T. Here is G, here is C. So they are form the bond between them, the one strand. Okay, so this is the structure of DNA and RNA. One remaining part, that is the based on this position of centromere in chromosome, they are further categorized into four types. What are they? This position of centromere. So there are mainly 4 types. Number 1, number 2, number 3, and number 4. Number 4. So there are 4 types according to the location of the centromere. This is the centromere. So when centromere is present in end of the chromosome, this is known as telocentric chromosome. Okay, this is telocentric. When it is present just below to the or near the end, this is known as acrocentric chromosome. When the centromere is present near the middle region, this is known as submetacentric chromosome, and when it is present in the center or middle of the chromosome, this is known as metacentric chromosome. Okay, so according to the position of the centromere, the chromosome are divided into 4 categories. To the position of centromere. This is the centromere. Okay, so these are the 4 types of chromosome according to their centromere position. So that's all for the today's video. If you like to get more such informative video and live classes, please do subscribe our YouTube channel Edwins, and also please do follow, share and comment in our Facebook page Edwins. Thank you, and please stay with us for the next part.